Hello everyone. Now we are going to learn about a topic related to molecular biology, DNA replication. So in this chapter you will learn about the individual events that occur at a replication fork and how these individual events together accomplish the task of DNA replication, the functions of enzymes and other proteins present at the replication fork, the different types of DNA replication used in eukaryotes, prokaryotes and viruses. So let's learn about this topic. Genetic information is transferred from parent to progeny organisms by a faithful replication of the parental DNA molecules. In most organisms, the information resides in one or more double-stranded DNA molecules. Exceptions do, however, exist. Some bacteriophage species contain single-stranded instead of double-stranded DNA. In these systems, replication consists of several stages in which single-stranded DNA is first converted to a double-stranded molecule, which then serves as a template for synthesis of single-stranded strands identical to the parent molecule. So genetic information is transferred from parent to progeny organisms by a faithful replication of the parental DNA molecules. We know that. So in most organisms, the information resides in one or more double-stranded DNA molecules. Exceptions do, however, exist, like some bacteriophage species contain single-stranded instead of double-stranded DNA. In these systems, replication consists of several stages in which single-stranded DNA is first converted to a double-stranded molecule, which then serves as a template for synthesis of single strands identical to the parent molecule. So from a purely abstract perspective, DNA replication might be thought to be relatively uncomplicated. After all, as proposed by scientists, each of the four deoxynucleotide bases need only be arranged so that they pair with an appropriate partner base, adenine with thymine, cytosine with guanine. Thus, we might expect that a gradual unzipping of DNA would allow nucleotide bases to fit in the other side and thereby replicating DNA quickly and effectively. Replication of double-stranded DNA is, however, a complicated process that is not yet completely understood, but still we are trying to learn about this. This complexity is at least in part due to the following features of the process. The two strands of a double-stranded DNA molecule run in opposite or anti-parallel directions. We know that. New nucleotides need to be added in such a way as to maintain those reverse polarities. New nucleotides need to be added in such a way as to maintain those reverse polarities. Replication requires a supply of energy to unwind the helix. Single-stranded DNA tends to form inter and intra-strand base pairs when it finds a complementary sequence, so that once unwound, separated strands of double-stranded DNA are likely to quickly reanneal, and a single enzyme can catalyze only a limited number of physical and chemical reactions. Since several reactions are required for DNA replication, a collection of enzymes is involved. Replication errors occasionally occur and mechanisms need to exist to correct them, and both circularity and the enormous lengths of some DNA molecules impose physical constraints on the replicative system. If those complications aren't enough to battle with beginning student of molecular biology, one more can be mentioned. Throughout the biological kingdom, some variations in key steps in DNA replication have been recognized. So our strategy for learning about DNA replication will be to proceed stepwise with a review of the important features of each individual step. First, we will review how twisted and sometimes circular DNA is unraveled prior to replication. Then we will learn that DNA replication usually begins at special sites on DNA. Next, we will study how the double helix is unwound to generate a single-stranded template. Later, we will discuss the enzymology of the replication process, which is complicated, as mentioned in the introductory remarks of this chapter, by the fact that the two DNA strands run in opposite directions. Finally, we will learn how all those systems and components are integrated into the so-called replication fork, the site on unwinding DNA where most of the action occurs. Then, semi-conservative replication of double-stranded DNA. That's the main topic. DNA replication is accomplished by semi-conservative replication. In this replication mode, each parental single strand is a template for the synthesis of one new daughter strand. The enzyme DNA polymerase adds nucleotide monomers one by one to the growing strand. As each new strand is formed, it is hydrogen bonded to its parental template. Thus, as replication proceeds, the parental double helix unwinds and then rewinds again into new two double helixes, each of which contain one original parent strand and one newly formed daughter strand. Then, twisting of highly coiled DNA is required for DNA replication. For the various replication enzymes to add nucleotide bases to the growing strands, it is necessary that the replication portion of a DNA molecule exists momentarily in the ladder configuration. As you learnt, double-stranded DNA is twisted or coiled 360 degrees for every 10 bases. This twisting must first be uncoiled so that the hydrogen bonds holding the two strands together are accessible to a special enzyme helicase. You may encounter it later in this chapter that unwinds the two DNA strands from each other. Untwisting of prokaryotic DNA is complex because they are circular and therefore have no free ends that might facilitate easy uncoiling. Also, DNA is usually highly folded and into compact structure so as to fit conveniently into a cell. Eukaryotic DNA, although long and linear rather than circular, is similarly twisted and tangled when it is organized into individual chromosomes. This twisting and tangling of DNA, overlooked in the simplistic diagram, presents a formidable task for cell preparing to replicate DNA. So, next topic, replication of circular DNA introduces twists. The circular structure of replicating DNA resembles the Greek letter theta. The term theta replication is therefore used to describe them. So, the circular structures of replicating DNA resembles the Greek letter theta. So, that's why there is, that term theta replication is used to describe them. So, replication of such circles involves a topological problem, a problem common not only to prokaryotic DNA, which is usually circular, but also to eukaryotic DNA. Although not circular, eukaryotic DNA is replicated simultaneously in many local regions, resembling long chains of small circles. Therefore, it is usually discussed in conceptual terms as if it replicates as a series of connected circles. As replication of the two daughter strands proceeds along the double-stranded DNA helix, in the absence of some kind of swiveling, the non-growing ends of daughter strands would cause the entire unreplicated portions of the molecule to become overwound or tight. This in turn would cause positive supercoiling of the unreplicated portion. This supercoiling obviously cannot increase indefinitely because if it were to do so, the unreplicated portion would become coiled so tight that no further advance of the replication fork would be possible. Most naturally, occurring circular DNA molecules are negatively supercoiled. Thus, the overwinding motion initially is no problem because it can be taken up by the underwinding already present in the negative supercoil. However, after about 5% of the circle is replicated, the negative superhelicity is used up and the topological problems arise. Next topic, special untwisting or uncoiling enzymes, topoisomerases exist. Separating the tightly twisted DNA requires that the individual strands are rotated about one another. A pair of topoisomerase enzymes accomplish this. A type 1 topoisomerase working just ahead of the replicating DNA marks a cut in one strand of the double-stranded DNA with the loose DNA ends attached to the enzyme. One of the single strands is passed through a gap generated by the cut. Then the ends is reattached to one another. As a result, a circular double-stranded DNA molecule is uncoiled with each topoisomerase action, one full 360-degree turn. As a result of this action, the DNA is relaxed somewhat. Later, the helicase enzyme mentioned previously will come into play and undo the hydrogen bonds and hold the 10 base pairs present in each full 360-degree turn. That is, topoisomerase works ahead of the helicase, revealing the overwinding mentioned. Then next, two topoisomerases function in concert. As DNA replication continues around a circle, positive supercoiling of the entire circle occurs. A second enzyme, which is also a topoisomerase, acts to introduce negative supercoils into the replication circle. It is called gyrase. This enzyme acts by cutting both strands, in contrast to the type 1 enzyme, which cuts only one strand. Therefore, it is also sometimes referred to as a topoisomerase 2. A segment of the cut DNA, which is located away from the actual cut site, is passed through the gap and then the cut is resealed. In this way, the replication circular DNA is maintained in its normal state of negative supercoiling. Next topic, initiation of DNA replication. Now that we have considered the problems associated with untwisting DNA in order to relieve unwanted supercoiling, let us examine the process of initiation. Initiation occurs at specific base sequences which comprise the so-called origin of replication, ORI, origin of replication. Two general types of initiation process, both acting at the ORI, have been discovered. One is referred to as de novo, from the new, in which synthesis begins from RNA primers. It is common to most organisms. The other is called the covalent extension mechanism. First, de novo method, or de novo in the sense from new, which is begins from RNA primers. Then there are another ways like covalent extension mechanism will be there. Covalent extension mechanism. It involves the synthesis of new strand as an extension of a pre-existing old strand. Its use is mostly limited to certain viruses. Those two general mechanisms for initiation are very important. Covalent extension mechanism and de novo mechanism. First, we are going to learn about de novo initiation. The most common mechanism of initiation, or de novo, is shared by DNA molecules as diverse as the E. coli chromosomes, phage lambda DNA, and the DNA of the eukaryotic simian virus 40, SV40, during the initiation of replication of the E. coli chromosomes. The DNA protein binds to a 9 nucleotide sequence that is repeated 4 times within a 250 nucleotide sequence. That region is defined as the ORI C. C for chromosomal. In the presence of ATP, the bound 50 kDa DNA protein monomers interact to form a complex protein DNA structure consisting of 50 to 20 DNA monomers and about 150 base pairs of DNA. Formation of this structure acts so locally denature an A plus T rich region directly adjacent to it. Then, because of specific interactions between the DNA A protein and other replication proteins like DNA B, the replication fork machinery is recruited to the unwound site on the DNA. Once all the requisite enzymes are present and primers have been synthesized, DNA pole 3 can begin to synthesize DNA. In the case of ORIC, two replication forks then proceeds in opposite directions away from the origin. The exact mechanism for initiation of the leading and lagging strands is somewhat unclear and currently under investigation. During stage lambda replication on E. coli, as well as the replication of the animal virus SV40, a very similar mechanism is used, and a virally encoded protein binds repeated sequence at the ORI to form an initial complicated nucleoprotein structure. This the mechanism seems to be general. A different type of de novo initiation is employed by other organisms. In this mechanism, an RNA primer is synthesized on one DNA strand through the direct action of an RNA polymerase enzyme. The eventual extension of that RNA primer by DNA polymerase during the synthesis of a daughter strand generates a replication bubble that consists of one double-stranded and one single-stranded branch. Since the growing daughter strand displaces the unreplicated parental strand, the bubble is called displacement loop, or more commonly D loop. Next, covalent extension initiation. In some of the very simplest biological systems like plasmids and viruses, natural selection favors rapid production of large numbers of copies of DNA. Using the covalent extension mode of replication, circular DNA molecules produce a concatama, which is a DNA base sequence. I'm not sure about the pronunciation. Base sequence tandemly repeated many times. In this mode of initiation, a nick is made in one of the DNA strands, thereby creating a free 3' OH end along with the free 5' P end. The free 3' OH provides DNA polymerase with the starting point. The synthesis of a daughter strand begins at this free end of the nicked DNA strand. That is, the free 3OH serves the same role as did the RNA primer illustrated for de novo initiation. A similar covalent extension process for the initiation of DNA synthesis is employed during the bacterial mating process. As we will be described earlier, a linear DNA synthesized is transferred from a donor cell to a recipient cell. Replication of virus DNA also employs covalent extension, and that's very important. Then next topic, unwinding of DNA for replication. In order for the enzymes that synthesize new daughter strands of DNA to begin their polymerization functions, the DNA template needs to be readied. This process of template preparation involves at least 3 steps: unwinding of the double helix, that the double helix is unwound 360 degrees every 10 base pairs, then breakage of the H bonds holding the two DNA strands together to expose the single-stranded templates for new synthesis, the separation of single-stranded DNA templates so that they will not re-anneal before DNA polymerase has had an opportunity to act. Although there is single one enzyme, DNA polymerase 1, that can accomplish all of those tasks. Its role in DNA replication is mostly limited to repair processes. Another enzymes, the DNA polymerase 3 mentioned earlier, is the main enzyme for polymerizing nucleotides in most cells. It is, however, highly specialized for polymerization and therefore requires the assistance of other proteins to carry out these steps mentioned previously. Then helicase enzymes unwind DNA. The unwinding of double-stranded DNA is accomplished by the helicase enzymes. When helicase encounter double-stranded DNA, they continue to move forward by unwinding and prying open the base pairs. The unzipping action of helicase involves breaking hydrogen bonds and eliminating hydrophobic interactions. Energy is required for this process. It is supplied by ATP, which is hydrolyzed in the process, and the energy released is used by the helicase enzyme. As mentioned previously, the single-stranded DNA exposed by the action of a helicase enzyme could easily reform hydrogen bonds with other bases on the opposite strand. This is prevented by the action of single-stranded DNA binding proteins, or SSB, which bind to the exposed strands, coating them in their single-stranded form and thereby blocking the re-annealing process. Once this strand has reacted with SSBs, polymerization of nucleotides proceeds. The unwinding action of helicase and the binding of SSBs is very important. Then next topic, elongation of newly synthesized strands. The enzymatic synthesis of new daughter DNA strands is complicated by 3 additional factors: the need for high fidelity in copying the base sequence, the fact that 2 parental strands are antiparallel, one runs 5' to 3' OH, and other 3' OH to 5' P. And DNA polymerase can only function in one direction. Third, the need for speed. The replication forks moves at 1000 nucleotides per second in E. coli. Considering those considerations, it is easy to understand that the number of steps that must be completed is far too great to be accomplished by a single enzyme. In fact, 20 or more proteins are not to be necessary. If in an effort to provide some understanding of the process with a minimum of confusion, each step will be treated separately. We will consider the basic chemistry of polymerization and the enzymes involved, the problems raised by the chemistry of polymerization, the mechanisms for eliminating replication errors, the synthesis of RNA polymers, and finally the topology of a complete replication apparatus. First, the polymerization reaction and the polymerase. In 1957, Arthur Kornberg showed that in extracts of E. coli, there exists a DNA polymerase, now called polymerase 1 or POL1. This enzyme is able to synthesize DNA from 4 precursor molecules, namely the 4 deoxynucleotide 5' triphosphates, DNTP, DATP, DGTP, DCTP, and DTTP. As long as a DNA molecule to be copied, a template DNA is provided. Neither 5' monophosphates nor 5' diphosphates nor 3' mono, di, or triphosphates can be polymerized. Only the 5' triphosphates can serve as substrates for the polymerization reaction. Soon we will see why this is the case. Some years later, it was found that POL1, although playing an essential role in the replication process, is not the major polymerase in E. coli. Instead, the enzyme responsible for advance of the replication fork is polymerase 3 or polymerase 3. Polymerase 3 also exclusively uses 5' triphosphates as precursors and requires a DNA template before polymerization can occur. This POL1 and POL3 have some features in common. The overall chemical reaction catalyzed by both DNA polymerases is polynucleotide 3' OH plus DNTP polynucleotide in plus 1 minus 3' OH plus PP, in which PP represents pyrophosphate cleaved from the DNTP. So the number 1 and 2 in polymerase 1 and 3 refer only to the order in which the enzymes were first isolated, not to their relative importance. Another polymerase, POL2, has also been isolated from E. coli. It plays no other role in DNA replication. Although mutant cells lacking POL2 grow normally, recent studies indicate that the enzyme may be involved in certain DNA damage induced DNA repair mechanisms. Additional DNA polymerases also implicated in DNA repair mechanisms have also recently been discovered. For convenience, the abbreviation POL will be used in this session and also in coming sessions. Then next major point, both POL1 and POL3 only polymerize deoxynucleoside 5' triphosphates and can do so only while copying a template DNA. Furthermore, polymerization can only occur by addition to a primer, an oligonucleotide hydrogen bonded to the template strand and whose terminal 3' OH group is available for reaction, that is a free 3 OH group. The meaning of a primer is made clear, and which depicts 6 potential template molecules. Of this, only 3 can be said to be active, each of which has a free 3' OH group. The lack of activity with D and the direction of synthesis with E and indicate that nucleotides do not add to free 5' P group. The lack of any synthesis with indicates an addition of 3' OH group cannot occur if there is nothing to copy. Thus we draw two conclusions. Both a primer with a free 3' OH group and a template are needed. Polymerization consists of reaction between a 3' OH group at the end of a growing strand and incoming nucleoside 5' triphosphate. When the nucleotide is added, it supplies another free 3' OH group. Since each DNA strand has a 5' P terminus and a 3' OH terminus, strand growth is said to be proceed in the direction of 5' to 3' direction. These 2 points are summarized. So occasionally polymerases add a nucleotide that cannot hydrogen bound to the corresponding base in the template strand. This may be purely a mistake or may result from the tautomerization of adenine and thymine discussed. So in any case, it is important that the unpaired base are removable while its incorrectness is recognizable, namely as an unpaired base at the 3' OH terminus of a growing strand. Then some other topics like correcting mismatched bases. Both pole 1 and pole 3 respond to an unpaired terminal base by terminating polymerizing activity. That is because those enzymes require a primer that is correctly hydrogen bonded. When such an impulse is encountered, a 3' to 5' exonuclease function, which is a separate catalytic activity located at a site different from the active site for nucleotide polymerization, is stimulated and the unpaired base is removed. After removal of the base, polymerizing activity resumes and chain growth therefore continues. This enzyme activity, because it removes a base from the end of a nucleic acid, is referred to as an exonuclease activity. This pole 1 and pole 3 are said to possess a proofreading and editing function. In addition to function of DNA polymerase 1 is that of an opposite direction. 5' to 3' exonuclease. The activity has the following features. Nucleotides are removed one by one from the 5' P extreme terminus only. That is, of course, the opposite of the activity, and more than one nucleotide can be removed by successive cutting. The nucleotide removed must have been base paired. The nucleotide removed can either of deoxy or ribo type. Activity can occur at a nick as shown. There are 5' P group. Then we will see shortly that the main function of the 5' to 3' exonuclease activity is to remove ribonucleotide primers. The 5' to 3' exonuclease activity at a single strand break or nick can occur simultaneously with polymerization. That is, as a 5' P nucleotide is removed, a replacement can be made by the enzyme's polymerizing activity. Since pole 1 cannot form a bond between a 3' OH group and a 5' monophosphate, the nick moves along the DNA molecule in the direction of synthesis, having a gap. This movement is called nick translation. So next topic we will be learning tomorrow. Thank you for listening. Next topic is about DNA polymerase 3 consists of multiple subunits. Then DNA polymerase can work only in the 5' P to 3' OH direction. These are the major topics associated with this, which we will be learning tomorrow. Then antiparallel DNA strands and discontinuous replication. Then Okazaki fragments, how they are joined. Then topics related to a primer is required for chain initiation. And some topics related to DNA ligase joins precursor fragments, the complete DNA replication system, then replication in the covalent extension mode, then bidirectional replication speeds up DNA synthesis, then replication of eukaryotic chromosome enzymology of human DNA replication, and some points which shows importance of this topic in practical sessions or a future practical application of this topic. We will be learning about all those tomorrow. Thank you for listening.